Hello students. In today's lecture, I will be discussing about a very important concept of regeneration, that is compensatory regeneration in mammalian liver. But before I tell more about the topic, I would like to mention that this presentation is a part of initiative taken upon by Zoological Society of Assam. Before starting the discussion, I would like to mention about the contents of the topic. It includes about the history of regeneration capacity, second, structure of liver functional units, that is the lobules, third, what are the cells involved in liver regeneration, fourth, modes of liver regeneration, fifth, regulatory signaling pathways of liver regeneration. At the very beginning, I would like to bring your kind attention that liver regeneration is an ancient truth. When we look back into the history of regenerative capacity, we remember about the Greek mythology where Prometheus was punished by Zeus for his sin. Zeus chained Prometheus to the rock and sent his eagle every day to feed on the liver of Prometheus. Amazingly, it was observed that the liver of Prometheus grew to its original volume, and the eagle was able to eat it the next day also. Furthermore, in the 19th century, liver regeneration is defined and described for the first time when a restoration of the liver tissue was noticed after removing a small part of the liver. Liver regeneration is the process by which the liver is able to replace lost liver tissue. The liver is the only visceral organ that possesses the ability to regenerate after partial resection and chemical injury. This beautiful feature is technically described as a compensatory hypertrophy, that is increase in cell size, followed by hyperplasia, that is increase in cell number of remaining hepatocytes to compensate the lost tissue. It is important to state that this process is neither a new creation of the missed tissue nor a true anatomical regeneration because it does not follow the steps of the true regenerative process, particularly the blastema formation observed in the epimorphic regeneration. Therefore, this phenomenon is termed as compensatory liver hyperplasia or liver regeneration. There are two events in which the liver has the capability to regenerate, one being by partial hepatectomy and the other being damage to the liver by toxins or infections. So, we can say mammalian liver regeneration appears to have two other lines of defense mechanism. First line of defense mechanism consists of normal matured adult hepatocytes which usually do not divide but are instructed to rejoin the cell cycle and proliferate until they have compensated the missing part. Second line of defense mechanism is activated only when adult hepatocytes are unable to regenerate well due to senescence, alcohol abuse, or diseases. In this defense mechanism, the quiescent population of hepatic progenitor cells are activated. The liver is organized from many lobules, which constitutes its functional units. Each lobule is composed of a central vein from which hepatocyte cords radiates towards the portal triad. The portal tride consists of the portal vein, bile duct, and the hepatic artery. The hepatic cords are single sheets of hepatocytes separated by sinusoids that carry blood from the portal trides to the central vein. This is an overall view of the position of the liver cells within the lobule. Within each lobule are a number of sinusoids, which are discontinuous vessels built from specialized fenestrated endothelial cells of the liver. Stellate or Ito cells are located in the space of Dc between the hepatocyte cords and the sinusoids. Cooper cells, which are the specialized macrophages of the liver, also reside in sinusoids. Cholangiocytes are the epithelial cells lining the bile ducts. So, the important cells which are responsible for liver regeneration or play the most important role in liver regeneration are the hepatocytes, second is the stellate cells, third the Cooper cells, fourth the endothelial cells, and fifth the cholangiocytes. Now, we will discuss about the first line of defense mechanism which occurs during normal liver regeneration and partial hepatectomy. In normal liver regeneration, the five types of liver cells such as the hepatocytes, duct cells, fat-storing Ito cells, endothelial cells, Cooper cells become activated and begin to divide themselves. Each type retains its cellular identity and the liver regains its ability to synthesize the normal liver-specific enzymes necessary for glucose metabolism, toxin degradation, bile synthesis, albumin production, and other hepatic functions. There are probably several redundant pathways that initiate liver cell proliferation and regeneration. Global gene profiling indicates that the end result of these pathways is to downregulate the genes involved in the differentiated functions of the liver cells while activating those genes committing the cells to mitosis. There are a number of pathways that initiate liver cell proliferation and regeneration. The liver removal or injury is sensed through the bloodstream. Some of the liver specific factors are lost while others are increased, such as the bile acids and the gut lipopolysaccharides. Bile acids activate the transcription factor FXR, which promotes cell division, while lipopolysaccharides activate hepatocytes to re enter the cell cycle. Cooper cells secrete the interleukin 6 and the tumor necrosis factor alpha. Stellate cells secrete the hepatic growth factor or scatter factor and the TGF beta. Blood cells produce the hepatic growth factor and the Wnt2 protein. Now, I will discuss about the different phases of liver regeneration. During liver regeneration, hepatocytes pass through three important distinctive phases. These phases are the initiation phase, proliferative phase, and the termination phase. These phases are regulated and linked together by multiple signaling pathways. Now first we will start discussing about the initiation phase. The initiation phase is also termed as the priming phase. It refers to the actions that occur in the early period of 0 to 5 hours after partial hepatectomy. These events, which are mediated simultaneously by cytokines and growth factors, modify the expression of multiple genes to prepare the entry of liver cells into the cell cycle. The earliest events of the initiation phase are the hemodynamic changes and increased activity of urokinase plasminogen activator. Partial hepatectomy causes an increase in the portal blood flow, resulting an increase in the availability per hepatocyte of growth factors and cytokines coming from the intestine and pancreas. A rise in the portal blood pressure is ensured, resulting in turbulent blood flow that exerts a mechanical stress on the endothelial cells. Endothelial cells express an increased activity of urokinase plasminogen activator following a mechanical stress. The urokinase plasminogen activator activity is observed 5 minutes after partial hepatectomy. Within 10 minutes, it prompts the conversion of plasminogen into plasmin, which breaks the fibrinogen into fibrinogen degradation products. The transformation of pro-matrix metalloproteinase into active metalloproteinases is augmented by plasmin at 30 minutes and lasts for 24 to 48 hours after partial hepatectomy. Both matrix metalloproteinases and plasmin are involved in matrix remodeling and turnover of many proteins of extracellular matrix. Remodeling of the extracellular matrix can initiate signaling impulses leading to release of locally bound growth factors that participate in liver cell proliferation. During matrix remodeling, an inactive hepatic growth factor attached to the extracellular matrix is activated to its active form by urokinase plasminogen activator and excreted locally and systematically, causing an activation of hepatic growth factor receptor or CMET within 30 minutes to 1 hour after partial hepatectomy. At the same time, an epidermal growth factor receptor is also stimulated by epidermal growth factor produced by the Brunner's gland of the duodenum and constantly goes to the liver via the portal route. Several studies have shown that there is cross interaction between CMAT and epidermal growth factor receptor, in which CMAT activation augments the activity of epidermal growth factor receptor. During the initiation phase, a number of regulatory signaling pathways are activated. These pathways are activated by external signals which are received by the Kupffer cell and transferred to hepatocyte, leading to liver cell proliferation. It is thought that innate immunity is the main stimulator for the initiation of liver regeneration. Following partial hepatectomy, activation of innate immunity takes place, leading to the production of its components such as the lipopolysaccharides and complement proteins, which binds to their respective receptors, toll-like receptors and the complement protein receptors in the surface of the Kupffer cell. This interaction leads to the stimulation of an important signaling pathway, that is the NF-kB pathway or the nuclear factor Kb pathway. When the external signals like lipopolysaccharides and complement proteins stimulate Kupffer cells, it excites the transcription of tumor necrosis factor interleukin 6. Tumor necrosis factor alpha plays a vital role in the initiation of two important functions. First is the activation of the NF-kB pathway. Second is the stimulation of a stress-activated protein kinase or C-Jun N-terminal kinase in hepatocytes. Stress-activated protein kinase or C-Jun N-terminal kinase are special kinase proteins that act as enhancers of liver regeneration. Interleukin 6 is also considered as a critical component in priming the hepatocytes for proliferation. Tumor necrosis factor alpha is also received by a receptor in the Kupffer cell, that is the TNF receptor, which leads to the activation of a signaling pathway in the hepatocyte, that is the Wnt beta-catenin pathway, which leads to the proliferation of the liver cells. This pathway is activated 1 to 3 hours after partial hepatectomy. Notch pathway is one of the earliest pathway that is turned on within 15 to 30 minutes after partial hepatectomy, which regulate liver regeneration. It is generally depend on two main proteins, that is the Notch 1 receptor and the jagged 1 proteins, which are upregulated 1 to 5 days following partial hepatectomy. In normal adult liver, Notch transmembrane receptors are expressed on hepatic progenitor cells, hepatocytes, cholangiocytes, and endothelial cells lining the portal tract vessels and sinusoids. They are stimulated by interacting with specific membrane-bound ligand proteins. All the signaling pathways initiate the transcription of the cyclin D and cyclin B in the hepatocytes. This leads the quiescent hepatocyte to be activated and re-enter the cell cycle for proliferation. The second phase of liver regeneration is the proliferative phase. The proliferative response to partial hepatectomy is divided into two main periods. Number 1, the period of induction. Number 2, the angiogenic phase. The period of induction refers to the proliferation of hepatocytes and cholangiocytes. The regenerative response of hepatocytes proceeds and triggers the expansion of cholangiocytes. These steps commences at the end of the priming phase and lasts for 72 hours, reaching the peak at 24 to 36 hours after partial hepatectomy. The second phase, that is the angiogenic phase, in which the non-parenchymal cells such as the hepatic stem cells, Kupffer cells, hepatic endothelial cells undergo proliferation in response to signal derived from proliferative hepatocytes. These last for 2 to 3 days. From the above discussion, we can conclude that the different factors such as the cytokines, growth factors produced by the endothelial cells, Kupffer cells, stellate cells allow the hepatocytes to divide by preventing apoptosis, repressing cyclin inhibitors such as P27 and activating cyclin D and E. Following the proliferative phase, the remnant liver tissue expands and retrieves the original size equivalent to the normal liver. The liver must grow through a. Now, we will discuss about the second line of defense mechanism, that is regeneration of diseased or damaged liver. The reproducibility of the liver after two-thirds partial hepatectomy is merely dependent on the proliferative features of the remaining parenchymal and non-parenchymal liver cells. All remnant hepatocytes enter one or two cell cycles to restore the original liver mass with very minimal engagement of the hepatic progenitor stem cells. Hepatic progenitor stem cells take the lead of the liver regeneration process when the mitotic capacity of the liver cells is restricted and overwhelmed as a result of severe liver injury. Hepatic progenitor stem cells differentiate into mature hepatocytes in circumstances associated with severe hepatocyte damage, such as acute hepatitis, chronic viral hepatitis, alcoholic liver cirrhosis, and massive liver resection. Hepatic progenitor stem cell or HPSC normally reside in the most distal parts of the intrahepatic biliary tree known as the canal of Herring. These canals form a normal connection between the biliary ductules in the portal tracts and the bile canalicular system in between the hepatocytes. The hepatic progenitor stem cell niche is unique microenvironment comprised of variable cell types such as the hepatocytes, then cholangiocytes, hepatic stem cells, endothelial cells, macrophages, and other inflammatory cells, growth factors, and cytokines. They are regarded as dynamic stem cells that persistently switch their differentiation patterns and expression of their markers according to the type of cell injury and which epithelial cells are subjected to most of the damage. For instance, in hepatitis, the primary damaged cells are the hepatocytes. Therefore, hepatic progenitor stem cells will be activated to enter the pathway that produce the hepatocytes. In contrast, if the cholangiocytes are the main injured cells, as in diseases like primary biliary cirrhosis, differentiation of hepatic progenitor stem cells into biliary epithelial cells will take place. During acute liver injury, the liver cells, hepatocytes, and cholangiocytes undergo acute cell necrosis. The necrosis debris from damaged hepatocytes are engulfed and removed by Kupffer cells. This phagocytosis stimulates Kupffer cells to secrete an essential cytokine called as the T and K-like weak inducer of apoptosis, which binds to its specific highly expressed receptor FGF inducible 14 on the surface of hepatic progenitor stem cells. This excites an important intracellular signaling pathway, that is the NFKB, leading to hepatic progenitor stem cell proliferation. The hepatic progenitor stem cells or the liver progenitor stem cells are the oval cells which are small progenitor cell population that appear to be in reserve and become active only when hepatocytes are unable to regenerate. This liver cell proliferation as a result of signal activation, they will differentiate into their respective cells, that is hepatocyte or cholangiocyte, according to the injury of the liver. From the overall discussion, we can say that in normal liver regeneration or partial hepatectomy, the hepatocytes are activated by various regulatory signaling pathways to enter into cell cycle for proliferation and differentiation. However, in diseased liver, the hepatocytes are not activated. Here, the hepatic progenitor stem cells are activated to undergo proliferation and differentiation into hepatocytes and cholangiocytes for liver regeneration. Here, I have cited some of the important references of the topic through which you can go through for better understanding. At last, I would like to thank you all for your patience hearing. For any feedback, please contact me at my email banasrimes@guwahati.ac.in.